Recently there has been a paper published in Blood showing the crystal structure, or the 3D structure of factor VIII incorporated in the factor VIII binding fragment of willebrand factor, the D'-D3 region. And by doing that, they've been able to not only see how the factor VIII is actually folded into this D'-D3 fragment, but they've also been able to identify the residues that are really in contact with each other during this in this interaction. And the interesting thing is that it's not only what we always thought, the C and N-terminal part of factor VIII that is in contact with the D'-D3, but also in the A3 domain there is a region that interacts with D'-D3. So the biosynthesis of factor VIII and willebrand factor has always been a bit of a dispute. For willebrand factor it has been relatively easy. It has been discovered already a long time ago, knowing that the endothelial cells and the megakaryocytes are actually the sources of willebrand factor production. For factor VIII it has been different because there is mRNA present of factor VIII in a lot of cells, including hepatocytes, hematopoietic cells, and endothelial cells. And some recent studies by the labs of Dr. Montgomery and Dr. Ginsberg, they really have shown by using some elegant mouse models that it's actually the endothelial cells that are the main source of factor VIII. Doesn't mean that they are always present in the same cells. That depends. In some organs it seems to be true. For instance, in the lungs you can clearly see co-expression of factor VIII and willebrand factor in the vascular endothelial cells. In contrast, in the liver, if you make tissue sections of the liver, then you can stain for factor VIII and willebrand factor. And then you see that factor VIII is expressed in the sinusoidal endothelial cells, whereas willebrand factor is expressed mostly in the endothelial cells that cover the large vessels. So depending on the organ, there is co-expression or not. The functions of willebrand factor and factor VIII are not exactly the same. Willebrand factor is mostly involved in the recruitment of platelets to site of vascular injury. So it's important to form the first platelet plug at the site of injury. Whereas factor VIII is mostly involved in is actually a cofactor for coagulation factor IX. So it's important for the formation of the fibrin network that stabilizes this platelet plug. We know that the level of factor VIII is about 100-fold lower than that of willebrand factor. So if there is any effect of factor VIII on willebrand factor function, then probably it's limited because of the molar difference between the two. But it has been shown by the lab of Long Zheng that if factor VIII is present, then it may accelerate inactivation of willebrand factor by AMTS13. That does not mean that if there is no factor VIII at all, that there will not be any cleavage of willebrand factor by this enzyme. But it seems that factor VIII is able to catalyze deproteolysis by AMTS13. Issue of light chain cleavage in respect to factor VIII function, it's not completely clear right now, because there are two issues that play together. On one hand, there is the acidic fragment that is released of the factor VIII light chain. And that release can have two functions. It can activate the light chain into another conformation that facilitates interaction with its cofactor factor IXA. But on the other hand, we also know that release of this acidic fragment results in dissociation of willebrand factor. And when willebrand factor is present, then it actually interferes with factor VIII binding to factor IXA, to phospholipids, etc. So whether the cleavage of the factor VIII light chain is necessary only to release willebrand factor, or also to activate the light chain into an active conformation, that we are not completely sure. For a long time we are interested in clearance of factor VIII, and one of the first things that you would like to do is to identify which are the main determinants of factor VIII clearance. And it appears that willebrand factor is really the dominant determinant of factor VIII clearance. And there are three main arguments to support that. The first one is that if you calculate how much factor VIII is actually in complex with willebrand factor based on the kinetic parameters of this interaction, then you can calculate that more than 95% of your factor VIII at any moment is actually in complex with willebrand factor. Yeah, so that's argument one. The second argument is that if you inject both factor VIII and willebrand factor in a mouse model where there is no willebrand factor at all, then you start to stain for the cells that have taken up factor VIII and willebrand factor, then you can see that both proteins are actually ending up in the same cell. So willebrand factor carries factor VIII to its clearance cell. And the third, and that is maybe the most illustrating one, is that you can actually predict the half-life of factor VIII solely based by knowing the blood group, willebrand factor levels and willebrand factor propeptide levels. And if you use a specific algorithm, you can use those three parameters to calculate the half-life of factor VIII with an accuracy of more than 90% in more than 80% of the hemophilia patients. One of the things that you would like to do to better understand the clearance mechanism of factor VIII is try to identify what is the clearance receptor for this protein. And the first time that we were able to identify a clearance receptor for factor VIII was in the late 1990s, where we identified LDL receptor-related protein 1, LRP1, as a clearance receptor for factor VIII. We were very excited because we thought, okay, we have a clearance receptor, we make some factor VIII mutants, and then we can prolong the half-life of this protein. However, it appeared more complicated than that. One of the things was that actually LRP1 is not the only receptor for factor VIII. Right now there is a whole list of other receptors that have been identified, including Cyclex, the asialoglycoprotein receptor, SCARA5, and a series of other ones. And I'm sure that this is not the end of the list that is going to be discovered. So knowing that willebrand factor is the driving force for the clearance of factor VIII, one option to prolong the half-life of factor VIII is actually to prolong the half-life of willebrand factor. Well, there are several ways in which you can do that. For instance, you can put an pegylated tag to willebrand factor, and we have seen that if you do that, then the half-life of your willebrand factor is prolonged about 4 to 5 times. The funny thing is that if you inject that pegylated willebrand factor in complex with factor VIII, you actually do not see any prolongation of the factor VIII half-life. And that's a bit weird because you would expect it's in complex with willebrand factor, so it would also have a prolonged half-life. Well, the main reason to explain that you don't see any effect on factor VIII half-life, or very little effect, is that actually the interaction between factor VIII and willebrand factor is highly dynamic. That means that once your factor VIII is on your willebrand factor molecule, it can hop very quickly to another willebrand factor molecule. So what you get is that you inject the complex, but your factor VIII is redistributed to the endogenous willebrand factor very rapidly. And since the endogenous willebrand factor has a normal half-life, also your factor VIII will be cleared normally. Knowing that prolonging half-life of willebrand factor does not help to prolong half-life factor VIII, another option is to put, for instance, a pegylated tag on your factor VIII or an FC fusion protein. Well, those things are now in clinical development or are already in the clinic. The problem is that the prolongation of factor VIII in this way is fairly limited. It's about between 1.3 and 1.5 fold. And if you look for other proteins such as factor IX, you can easily reach 4 or 5 fold prolongation. So, to my opinion, the best way to prolong the half-life of factor VIII is to develop molecules that can circulate independently of endogenous willebrand factor. And there are a number of ways in which you can do. For instance, Biogen has developed a molecule in which the factor VIII is actually covalently linked to the D'-D3 fragment of willebrand factor, which binds factor VIII, plus an FC fragment. So in that way, your factor VIII cannot be redistributed to your endogenous willebrand factor. Another option is just forget about factor VIII and try to work on factor VIII bypassing agents like monoclonal antibodies or aptamers or scRNA, etc. Because monoclonal antibodies, they have very long half-lives of sometimes several weeks. So in that way you can prolong the half-life of your treatment very easily. So one of the issues that complicated the search for factor VIII molecules with a long half-life in the interaction with LRP1 was that under normal conditions, willebrand factor is able to block the interaction between factor VIII and LRP1. But one of the things that we noticed while using specific mouse models is that if you knock out LRP, you do not only get an increase in factor VIII levels, but also your willebrand factor levels go up. And by doing a number of studies, we actually have identified that not only factor VIII, but also willebrand factor is a ligand for LRP1. And the difference is that factor VIII is able to interact with LRP under static conditions, and willebrand factor can interact with LRP1 only under conditions of shear. So that means that the blood flow needs to be present for willebrand factor to interact with LRP. So in the end, it's not only factor VIII that interacts with LRP1, but also the factor VIII willebrand factor interacts with LRP1. So there is a whole range of scavenger receptors, including LRP1, that is able to remove factor VIII, willebrand factor, and the complex of both proteins.